I'm a systems biologist. I have a combination of a wet lab where we do experiments and a dry lab. It's called Vanaja Labs for now. My overall goal, a long-term goal, is to build computational and mathematical models of signaling networks that are implicated in diseases like cancer and diabetes. And then with these models, help predict where resistance can occur in cancer, and if we can come up with not exactly a cure, a better way of managing diabetes by understanding the computational models built of it. Right now, I have a small group with 2 postdoctoral fellows. We have 3 graduate students, and we have a collaborator who is my wife from the Memorial Sloan Kettering Cancer Center, who does some of the experimental work we collaborate with. To put it mildly, cancer has not been cured yet. There is a reason why it has not been cured, partly because life, as it has evolved on this planet, is almost 1 billion years old. So anytime we try to snuff out life, there are multiple ways in which life can get over the block. So that was one of the fundamental understanding I kind of had when I was starting this program, and I figured that any simple approach where if there is a mutation and then you go block the mutation in a cancer is not going to work. So we had to come up with a very cleverer way of trying to understand what these cancer cells were doing when they were inhibited by these drugs. And that is what sort of inspired me to come up with this newer way of proposing different therapeutics. I met my wife in grad school. She was a master's student. I was doing my PhD, but then she ended up doing a PhD in a breast cancer lab in Memorial Sloan Kettering Cancer Center in New York. And one of the sobering aspects of doing research in these institutions is that you often walk through hallways where patients are undergoing treatment. My wife, she's also a singer. She sings. She's a part of a band. So she signed up for 2 years of so MSK has this unit where kids who have terminal stage are you take care of kids who have terminal stage cancers. And they have a room where all the kids spend their time. And you have people who sing and play music come and try to, just make it easier for them. And so she volunteered for 2 years. And it took it takes a toll on a person when you see young people, they probably don't know, but we know. And then, you're trying to make that easier, but then you're sort of also helpless that, this is you're a professional who is trying to make a difference, yet you can't. And you walk through these hallways every day. So that kind of, just somehow imprints on you. You want to have whatever you do somehow aid in the direction that can help. Some of the intricacies involved in how we have designed this project requires that we have some sort of help and collaboration with the industry in multiple stages. So what this would entail is using all of the latest omics measurements, like say single-cell proteomics, single-cell ATAC-seq, single-cell RNA-seq, and then use a deep learning model to then learn the mapping between what would be a cellular phenotype, meaning cells migrating, cells dividing, cells acquiring resistance phenotypes, and then learn this mapping to predict when you have a newer tumor, how that tumor is going to behave when you input a certain therapeutic. So there are multiple stages involved where you need to develop a deep learning neural net, you need to develop a big data set of all possible mapping from measurements inside a cell to the phenotypes, and then go on to use it in clinical trials, clinics. So there are multiple stages, and collaborations from the industry is sort of required at every stage.